According to the American Cancer Society, in 2014, there was an estimated 14.7 million people living with cancer in the United States, and of that, about 400,000 people are living or in remission from leukemia. Cancer cells rapidly proliferate without stopping and invade into surrounding tissues, while normal cells divide to create new cells that the body needs. Hyperplasia and dysplasia are stages of proliferation that may lead to cancer. Leukemia is a form of blood cancer that starts in the cells, which would normally develop into different types of blood cells. It affects white blood cells, which are known to be infection fighters. In addition, leukemia alters normal growth and development of white blood cells. Currently, common treatment for cancer patients include chemotherapy, radiation, surgery, or a combination of three. Chemotherapy targets rapidly growing cells, which is one of the main characteristics of cancer. However, the caveat of that is that it kills cells that are rapidly growing in normal circumstances. These treatment options are not your only options. Now there's a new wave of targeted treatments. We now utilize a powerful diagnostic tool called sequencing, which can help doctors determine how a disease is unique to each patient and how to effectively treat them. Using this diagnostic tool, doctors and patients can use targeted therapies that are specific to each patient's disease. One such example where sequencing is used is in leukemia, and specifically acute myeloid leukemia. Uses this approach to identify important characteristics of the disease, which can vary from patient to patient.
DNA is the blueprint of a cell, and the order of these bases, known as nucleotides, is what makes a person unique. Sequencing is the decoding of a DNA molecule by determining the sequence of these nucleotides. Three key people who pioneered and developed the initial methods were Frederick Sanger, Walter Gilbert, and Alan Maxim. Two of these three earned the Nobel Prize in 1980 for their contributions in chemistry. First, Frederick Sanger developed his plus-minus method in 1975. Next, Walter Gilbert and Alan Maxim developed their chemical cleavage method in 1977. Finally, Frederick Sanger published his method called chain termination sequencing in 1977 as well. This was the most widely used method due to its advantages over the previous developed method. Sanger's chain termination sequencing was the method used in the Human Genome Project, which took place between 1990 and 2000. Their methods have proven to be incredibly valuable in how we understand human biology and have led to countless breakthroughs in biomedical and general sciences. Sequencing has come a long way from the methods used in the Human Genome Project. The commonly used methods today are referred to as next-generation sequencing, or NGS for short. A few of the most relied upon methods were developed or acquired by companies like Illumina, Applied Biosystems, and Thermo Fisher. These companies provide the most commonly used sequencing methods for clinical and research applications today. The specifics of whole genome sequencing vary from method to method, but they all determine the nucleotide sequence of a DNA molecule in some roundabout way. NGS methods are incredibly high throughput, which means they are now fast and cheap. This simply means that NGS can be used as a diagnostic tool for various diseases. Many diseases are caused by a mutation of a certain region of DNA, where a nucleotide is switched or deleted. An example of this is AML. NGS can find these mutations, which are unique and can vary from patient to patient. Determining the mutation a patient may have with NGS gives doctors and patients valuable information about how to treat their specific disease.
AML can be caused by an indel. An insertion is the addition of one or more nucleotide base pairs into a DNA sequence, and a deletion is defined by any number of nucleotides deleted. Therefore, indels can lead to incomplete or incorrect proteins.
Using whole genome sequencing, two main gene mutations were identified with those diagnosed with AML, IDH2 and FLT3. Two drugs have been developed for targeted therapy for AML. Those who have a mutation in the FLT3 gene, midostaurin works by blocking the FLT3 protein and several other proteins on cancer cells that aid in growth. And for those who have a mutation in the IDH2 gene, enasidenib works by blocking the IDH2 protein. This protein seems to work by helping leukemia cells mature or differentiate into more normal cells. By identifying a specific mutation, a patient's doctor can correctly propose a course of action, thereby significantly improving the patient's survival rate. We have come a long way from treating all types of cancer with one drug to various drugs treating specific diseases. Although there are ethical concerns, targeted therapy allows for personalized medicine wherein deleterious conditions can be clearly identified and treated effectively. Now, we can perform liquid biopsy, which increases sensitivity for smaller samples, and we are currently in the process of successfully developing third-generation sequencing.